Okay, thanks, Serena. So today I'm going to be talking about some work that my colleagues and I have been doing, looking at how mechanical loads influence cranial shape and function during development. Some of you know, most of you will know Jen, and then my other colleagues, Lucy, Karen and Chrissy, who work in Bristol in biomedical sciences. And we're interested basically in looking at what controls morphology. If we can look at this very broadly, there are two facets here. There's the genetic control, and then there's what I've classified here very simplistically as the environmental control. And environment can control morphology during adulthood, but it's most prevalent during development. And you can classify this down into a series of different kinds of effects here, which can all control the ultimate morphology of an organism. Things like the positional information of where the cells within the developing animal sit next to each other. Things like gravitational forces, particularly important for mammals that have embryos in utero. Cellular forces, things like the contact pressures and adhesion of different cells. And in particular, what we're interested in here, the effect of mechanical loading on shaping the skeleton. And I've deduced this down here into what we would think of as embryonic muscle contractions, and how do they influence the shape of the skeleton themselves. So we have quite a very broad understanding of how mechanical loads influence skeletal morphology. And this is kind of summarized by this diagram here, which builds on work that's from the late 19th century by people like Julius Wolff, which looked at the relationship between the loads a bone experiences and the ultimate shape it ends up being to deal with those kinds of loads. So this was modified in the 60s by a guy called Harold Frost, and it's named here as the Utah paradigm of skeletal physiology. But basically, in its very simple state, is that you've got this adapted state that the skeleton sits in when it's experiencing kind of a range of everyday strains. And when that's happening, just normal bone turnover occurs. But if the bones fall into a period of disuse, for example, and the strains decrease here, then we see bone being taken away where it's not needed. And conversely, if the strains increase, either in magnitude or frequency or both, then we get into a period here of overload and more tissue is deposited. So this is a nice theoretical example here, but there are many studies that have looked at this in animal models where bones have been incised or animals have been exercised or subject to immobilization, and it's shown how these criteria can work. We also have good examples from human studies as well, things like low or zero gravity or bed rest, where the bones fall into disuse, the strains are lower, and there's a problem with tissue loss. And then likewise, many examples from overload here or increased strains, for example, in humans that undertake strenuous activities, people who undertake high impact or endurance activities, particularly later on in life, can still remodel their skeleton. And then also interesting studies, because we have paired arms and legs, we can look at the effect of what happens when one of those arms or legs is doing something very energetic and the other arm or leg is not doing anything. And we can see how muscles increase and then bones increase in density and thickness to compensate. So there's a good idea there of what's happening in the adult form. We also have a nice idea about how mechanical loads can influence embryonic skeletal development, although we're really just only beginning to start to understand how this process actually works. And so, for example, we've got a mouse mutant here, which is lacking muscles of some of the postcrania, and then a control here. And as we proceed through development, we can see the skeleton in this dark blue here has abnormalities. The tail's all curved round. There are abnormalities in the head, and also eventually it's not fully ossified like the control form here. So lack of muscles means that the skeleton isn't developing properly. Furthermore, there's been recent studies. One of the people who's been leading this is Niamh Nolan and her lab at Imperial, and other people as well too, that have looked at linking the mechanics of the developing skeleton to skeletal formation and the expression of genes that may be mechanosensitive. So here we've got a couple of finer element models that are simulating normal flexion or extension of the tibiotarsal element of a developing chick. And then a chick tibiotarsal element here that's under rigid paralysis, so all the muscles are contracting at once. And what we find in these models is that the strains and the fluid velocities within the normal model where they tend to localize is where we see the expression of putative mechanosensitive genes, and it also tallies with where we see the ossification front. So this chick element here is proceeding through development here, and here's the ossification front here. The bone's starting to ossify in the center of the diaphyseal shaft, and it's proceeding towards the epiphyses here. So what our study is particularly interested in looking at is craniofacial development. And this is it's not largely been ignored, but it's more been a kind of a byproduct of studies that have looked mainly at the postcrania. But we're doing this by looking at disruptive development, either experimentally or genetically. And we're using zebrafish as the model system. And zebrafish look like this when they're adults. They're sort of 3 to 4 centimeters in length. They can live for a remarkably long period of time, about 18 months to 2 or 3 years in age. But what we're interested in, particularly for the studies I'm going to present to you here, are the larval stages here, very early post-fertilization stages. And you can see here, this fish is about 5 days old. It's got a mouth, it's got large eyes here on the side, and it's feeding from a yolk sac here. And down here at the bottom is a fish that's is 32 days old, so this will be a juvenile fish. It's been stained for muscles, and you can see is the adductor mandibulae, the jaw closing muscle, many of the other muscles of the head and the gill skeleton. But we're interested more in this age up here at the top. And this is 6 days post-fertilization. But what's interesting to see here is that many of the muscles, like the adductor mandibulae and some of these other muscles here sitting in the floor of the skull here, are already fairly fully formed. So just to orientate yourself in some of the images that I'm going to show you, before I started this work, I wasn't familiar with the embryonic zebrafish musculoskeletal anatomy, and I don't expect many of the audience are as well too. So what we're going to be focusing on looking at here are the 2 cartilaginous elements here, the palaeontology and the Meckel's cartilage. Now, forming the putative jaws here, but you can see they're not aligned like in a classic jaw formation. They're kind of separate and aligned against each other. But here's the adductor mandibulae muscle here that closes the jaws. In ventral view, you can see the Meckel's cartilage is bending round here, and here's the palaeontology and the adductor mandibulae. And we're also going to focus on the action of these muscles here, the intermandibularis anterior, which runs between the 2 Meckel's cartilages, and the intermandibular posterior, which runs from the Meckel's cartilage to the basihyal here. So these will be some of the images that you'll see later on. So why use zebrafish as a model system when they're derived teleosts? Well, basically they're very easy to breed and house, and they have rapid development as well too. We can look at live imaging as well too, so we can put the fish under a microscope and see what they're actually doing. There are many transgenic lines as well too, which fluoresce and label different features, like the collagen here in these developing cartilages. And there are many mutant forms as well too, which have different types of abnormalities. This one here that we're looking at here hasn't got any craniofacial musculature apart from the sternohyoideus right at the back. We can also do live lineage tracing as well, so look at how the cells actually change position and shape as the animal matures. So what we were looking at for this study then was we wanted to know when the muscle forces were initiated in those very early embryonic stages, and to look at the morphological differences in the joint and cartilage element shape in fish that have and don't have mechanical loading during development. So then drilling down into that, we looked at where muscle activity was crucial to producing a normal joint. And then we looked at the differences in strain environment between the normal and the developmentally disrupted joints, and then to see if that led to any differences in the morphology of the cells. So we could then actually look at the mechanism by which those mechanical loads might be affecting strain, which then might be affecting what the cells are doing. So in our experiments, we have some controls, and we have these mutants that don't have any muscles. And then we have these other mutants, these other normal fish here rather, which are subject to what's called flaccid paralysis. So they're essentially Botoxed, so their muscles are really loose. And then we have these rigid paralysis fish where the muscles are in contraction the whole time. And then we have this mutant here, which is kind of a hyperactive mutant, which thinks it's lacking oxygen, so it moves its mouth a lot more than a normal fish would do. And so then there's an extra mechanical stimulus. So in our models here, first of all, what we did was characterize when the jaw started moving. And you might be able to see in this little fish here, you can see the jaw moving just a little bit. This whole fish here has been embedded in a lump of agar here with its head poking out, so we can just measure how much its jaws are moving here. And then what we find in these, here's the hours post-fertilization. So these are really, really early stage fish. But when we get to 3 days, they start to move their mouths. So we know that muscle forces are kicking in them. But then between 3 and 4 days is when there's a big jump in how often or how frequently they open and close their mouths every minute. So what does that mechanical loading then do to the muscles and the cartilage? Well, it disrupts them, it messes them up. So here's the muscles, and we're looking in ventral view at our fish. And you can see I've put this little schematic here. And then here's the cartilage as well too. So here's our mutant that doesn't have any muscle, and indeed it lacks muscle. And you can see its cartilages are a very different shape. Here's our Botox fish with its very flaccid, wriggly muscles here. And here's our fish that's experienced this tight contraction the whole time. And then here's our hyperactive fish as well too, which has got more musculature and then a more robust Meckel's cartilage. So we quantified this by doing outline analysis of the jaw shapes. And you can see quite clearly here that there's a lot of difference here, which is supported statistically between the shape of the different Meckel's cartilage. So here's our mutants with no muscle. They've got very different jaw shape, a Meckel's shape to the control. And so have the hyperactive forms. Interestingly, the fish that have been anesthetized, whether it's flaccid or rigid paralysis, they're indistinct in the shape of those Meckel's cartilages. But basically, if you've got different mechanical loading environment, it leads to a different shape to Meckel's cartilage. So that's important for the shape of the jaws. Also as well too, and this is where my colleagues are particularly interested in this, because they're interested in arthritis and how mechanical loading might disrupt joint formation, is that when we look at our different types of fish, different mechanical loads disrupts the jaw joint. And it does it in quite a consistent way. So here's our normal jaw joint here, between the Meckel's and the Palaeontology, and there's a nice even distance at the interzone between those two joints there, and you can see that they meet quite neatly. In our mutant forms, what happens is on the medial side, the jaw joint grows over, and then on the lateral side, what happens, it grows away. So we've got this disrupted jaw joint shape as well too, when we either anesthetize the muscles, or even when the animal's moving its jaws too much, it also messes up its jaw joint. So there's kind of a sweet spot there in what they need to do. So we find that muscle movement's required for a normal shaped jaw and a normal jaw joint. And you need to be moving your muscles, not just having muscles there, because remember those rigid paralysis ones, they've got their muscles contracting, but those muscles are not moving and they still have messed up jaw joints. What we wanted to look at was exactly when this muscle loading was really important. So our fish are ranging from 3 to 5 days post-fertilization. And what we found was that the crucial time for having your muscles disrupted that affects your jaw shape is at 4 days. So here we've got our little blocks here where they're coloured in, means that an anaesthetic has been applied. So when that happens at 4 days, we get a messed up jaw shape. When it happens for longer, the jaw shape is more messed up. But 4 days seems to be really crucial to normal joint formation. If you don't have any muscles acting then, your joint and your Meckel's cartilage is going to develop abnormally. Also as well, we looked at the symphysis, and that's also messed up by the lack of mechanical loads. And what we find is in our mutants with no muscles and the ones which have been Botox, so their muscles are flaccid, we find that the cells at the symphysis are more circular, they're smaller, and they're orientated differently as well too. So, and you can see here the difference in shape and just sort of and formation of the symphysis compared to what we see in the control. Now, interestingly here, what happens is when the muscles are contracting but just rigidly, the symphysis looks okay. So what we've found is that you need that dynamic muscle contraction for normal shape of the Meckel's cartilage and normal jaw joint shape. But you just need some kind of muscle tension for normal symphysis development. So the signal is different for different parts of the jaw as well too. Now finally, we're interested in what happens to the mechanics of the jaw when the morphology is disrupted by this lack of muscle action. And to do this, we use finite element modelling to look at the strains within the developing cartilages. And our workflow is that we start off by building the finite element models from confocal images. These are really tiny fish, and we're down to sort of sub-micron level resolution here to actually build these models. But the beauty of this, rather than working with fossils, which I'm more used to, is the amount of data that we can actually input into our models. I'm not going to go into this in huge detail here, but we have detailed information. We can look at individual muscle fibers. There's only about 4 or 5 in a 5-day post-fertilization fish. There are measurements for the muscle stress of embryonic zebrafish muscle fibers. We can get cartilage material properties from atomic force microscopy studies. So we can get a really detailed picture of all the input parameters that we need to put into these models. We build them in a VISO, and then we analyze the models using the normal workflow that we use in the lab, which is through a Hypermesh and Abacus, 2 software packages that we use. And so for this study, we had 3 different simulations. So we had a simulation of jaw opening and jaw closing, with the different muscles contracting as they would normally. And then one to simulate rigid paralysis, so where all those muscles are contracting at the same time. And what we find is that we find differences in stress in the cartilages between jaw opening and jaw closing and rigid paralysis. But universally, the high areas of stress localize to these areas of abnormal morphology that we've spotted in our confocal and our other analysis. And that is in jaw opening at the symphysis, we get high stress. And then in jaw closure at the jaw joint, we get really high stress. And in our rigid paralysis models, both those regions are under high stress because all of those muscles are contracting at once. So if we look at what's happening at cross sections, for example, through the jaw joint, there's tensile strain at the lateral jaw joint during closure and compressive strain at the medial joint during jaw opening. And we had an MSc student who spent a long summer measuring the orientation of cells at different regions across those jaw joints here. And her Rose diagrams here show those different orientations. And what we found was that in the anesthetized fish and the mutant fish, the cell orientations change shape on this medial side of the jaw when the muscle load is reduced. So maybe this compressive strain is doing something here. And then also we looked at an indicator of cell maturity, and we found that when the muscles are absent, we see more cells in regions of high stress and strain, and they're less mature when those muscle forces are absent. So then to wrap up, what we found out is that the zebrafish start to move their jaws between day 3 and 4, but it's the loading between days 4 and 5 which is really important for normal jaw, a jaw joint, and symphysis development. And if you remove those muscles, then the shape and the morphology is altered. And you need different types of loading, so dynamic loading to maintain a normal jaw and jaw joint shape, but just any kind of muscle loads to maintain a normal symphysis. And these high strains and joint abnormalities are linked, and they may be linked to things like cellular states, so cell size and orientation, and the maturation of cells as well too. So then I thank my co-PI Chrissy here, Lucy and Karen and Jen and other people who've helped out. And if you are interested in looking any further, we've done a Journal of Visualized Experiments video here which shows our full work through. So some of the some of the lab and some of the modelling criteria as well too, so you can look and see exactly what we've done. Thank you very much.